The scientific method is a systematic approach used in scientific inquiry to explore observations, answer questions, and test hypotheses. It involves a series of steps that help researchers objectively observe, experiment, and form conclusions about the natural world. Here's a breakdown of the steps with examples. Observation. This is the first step where scientists notice something in the natural world that piques their curiosity. It could be anything from a pattern in data to a strange phenomenon. For instance, a scientist notices that plants near a window grow taller than plants placed further away from it. Research or background information. Scientists gather information and existing knowledge about the observed phenomenon to better understand it. This may involve reading scientific literature, consulting experts, or reviewing previous studies. For example, the scientist researches how light affects plant growth, looking at previous studies related to photosynthesis and plant biology. Forming a hypothesis. A hypothesis is an educated guess or prediction that attempts to explain the observation. It must be testable and falsifiable. For example, the scientist hypothesizes, if a plant receives more light, it will grow taller. Alright, so next up we have experimentation. The scientist designs and conducts an experiment to test the hypothesis. This involves using controlled variables and independent and dependent variables to ensure a fair test. For instance, the control group does not receive the independent variable, which in this case is light. The independent variable is the factor being changed, such as the amount of light. The dependent variable is the outcome being measured, like plant height. To give you a clearer picture, let's consider an example. The scientist grows two sets of plants, one in direct sunlight and one in a shaded area, making sure other conditions like water, soil, and temperature remain the same. Moving on, we have data collection. The scientist collects quantitative, which is measurable, and qualitative, which is descriptive, data during the experiment. This could include numbers, observations, or even photographs. For example, the scientist measures the height of the plants daily and records how quickly each group grows. Finally, we come to analysis. The data is analyzed to determine whether the hypothesis is supported or refuted. Scientists often use statistical tools to make sense of the data. For example, after measuring the plants, the scientist finds that the plants receiving more light grew 20% taller than the shaded plants. Statistical analysis shows a strong correlation between light and growth. Alright, so let's wrap things up with our conclusion. The conclusion, in essence, summarizes the findings of the experiment and states whether the hypothesis was correct or incorrect. For example, the scientist might conclude that the hypothesis was supported, saying, plants that receive more light do indeed grow taller. Reporting and peer review. The results of the experiment are shared with the scientific community. Other scientists may review the work, replicate the experiment, or critique the findings. For example, the scientist publishes the results in a scientific journal, detailing the methods, data, and conclusions. Peers review the work to ensure it meets scientific standards. Replication. Repeating the experiment is crucial to ensure that the results are not due to chance. Replication builds confidence in the findings. For instance, another researcher might conduct the same experiment in a different location or under different conditions to see if the results hold true. Real-world example, the discovery of penicillin. In 1928, Alexander Fleming noticed that a mold, Penicillium notatum, accidentally contaminating his bacterial cultures, killed the bacteria around it. Fleming reviewed previous studies on bacteria and molds to understand the potential of antimicrobial agents. He hypothesized that the mold produced a substance that could kill bacteria. Fleming isolated the mold, tested its effect on various bacteria, and confirmed its antibiotic properties. He collected data showing how the mold inhibited bacterial growth. Fleming concluded that the mold produced a compound, penicillin, that could be used to treat bacterial infections. Fleming published his findings, and later other scientists like Florey and Chain replicated and extended the work, leading to the mass production of penicillin. In sum, the scientific method ensures that scientific knowledge is based on objective, repeatable observations and experiments. It provides a framework for understanding the natural world and answering complex questions in a structured and reliable manner.